Good afternoon, one and all. Today I am going to give oral presentation on biodiversity of Calosobruca species Bruchidae coleoptera in Terai zone of West Bengal. First of all, topics I am going to cover are introduction, materials and methods, result, summary and conclusion, future scope. Introduction. Calosobruca species family Bruchidae belonging to order Coleoptera are well known as primary and most destructive pest of stored legume seeds. In India, these are commonly known as pulse beetle. Pulse beetle is cosmopolitan in nature and capable of attacking wide range of legumes, namely green gram, chickpea, pigeon pea, and lentils. This causes damage to the grains which were stored at temperature of 30 degrees centigrade and 70% relative humidity. Pulse beetle has emerged as one of the most serious insect pests of the different pulses, particularly in storage. Biodiversity of Calosobrucus is often neglected by many researchers by assuming that it has limited or no diversity. The information regarding this aspect is also scanty for this region of West Bengal. Keeping this in a view, a survey was conducted in three districts, that is Cooch Behar, Alipurdwar, and Jalpaiguri districts of Terai zone of West Bengal, to find the diversity and relative abundance of the pulse beetle in chickpea during 2018 to 20. Materials and methods. First of all, to know the diversity of pulse beetle in Terai zone of West Bengal, what I have did is I have collected 500 grams of chickpea seeds from different means five grocery shops of three districts: Cooch Behar, Alipurdwar, and Jalpaiguri. Then I have packed these seeds properly in district wise in the glass jars. Then I have kept it at a side until the emergence of pulse beetle, until the emergence of pulse beetle. Later, what I have did is I have collected the adults which were emerged, and have identified preliminarily under the stereosome binocular microscope by using the taxonomic key given by Khare in 1993 and Haxon 2016. Then it was sent to the taxonomist of NBAIR for identification and confirmation of species. Then for relative abundance of species, what I have did is, after emergence of insects in glass jar, randomly 100 insects were collected. Then these insects were separated and counted according to the species to study their relative abundance by using the following formula. This is the formula I have followed. The next result what I have got is, regarding this biodiversity, after observation of antennae, hind legs, and elytra, it revealed that the insects which were emerged in the glass jar were belong to genus Calosobrucus, family Chrysomelidae, subfamily Bruchina, and order Coleoptera. For genus identification, what I have the morphological characters what I have identified are, there are pronotum and hind femur. Here in pronotum, it will have double raised area in the middle at the base of the pronotum, and the area is covered with pale hairs. While in case of hind femur, it will have tooth on inner and outer surface. Then species identification. For species identification, a microscopic study of morphological parts like antenna, hind legs, sternite regions, and elytrum of the species of the genus Calosobrucus were done by using taxonomic key. Later, I have identified that the species were Calosobrucus chinensis, maculatus analis, and another species was also found under this genus, that is under this genus Calosobrucus, but the species name was up to now we didn't get what that name it is. Then next, morphologically different among the these are the morphological difference between the species I have observed. That is, first of all, antenna. Coming to antenna, in Calosobrucus chinensis, the female will have serrate type of antenna, while in case of male, it will have highly pectinate type of antenna. While in case of maculatus, we will find both in case of male and female slightly serrate type of antenna. While in analysis, male and female will have subserrate and testaceous type of antenna. Then in case of by considering lateral aspects, Calosobrucus chinensis will have densely patches of white chorus bristles on the side margin of the segments 2 to 5 of this ternite. Second to fifth segment, we will find the white chorus bristles like a setae, while in case of male and female both. While in Calosobrucus maculatus, we will find dense white patches of the setae on the caudal margins of the metaepisternum, both male and female. In case of analysis also, we will find dispersed white patches of setae, same on the caudal margin of the metaepisternum region. Then abdominal stenoid, in all the cases, in all the species, we will find in female the abdominal stenoid will be distinct and separate, while in case of male congested. Then hind femur, coming to hind femur, Calosobruchus chinensis female will have a tooth on outer and inner carina, while in case of Calosobruchus maculatus, the female will have the tooth on outer carina was larger and blunt, whereas a sharp tooth of same size is present on the inner carina. While in case of female, tooth on outer carina was largely pointed, while the tooth present in inner carina was very minute. Then Calosobruchus species indet. Here, how that insect emerged was belongs to genus Calosobruchus. How we identify is that by seeing the pronotum. By seeing pronotum, we got that it has double raised area. So, we concluded that this species belongs this insect belongs to genus Calosobruchus. But up to now, we didn't find what that species name is. So, that species name is index species, like that we have kept. Then relative abundance of Calosobruchus species. Relative here we got that Calosobruchus in Cooch Behar district, Calosobruchus chinensis was more abundant, while in case of Alipurduar district, Calosobruchus maculatus was high, while in case of Jalpaiguri district, Calosobruchus chinensis was recorded high. Summary and conclusion. Here I conclude that the in this whole study, I have got that four species of Calosobruchus were recorded on chickpea seeds in Terai zone of West Bengal, that are Calosobruchus chinensis, Calosobruchus maculatus, annalis, and Calosobruchus species indet unknown. Then coming to relative abundance, here that Alipurdwar, Calosobruchus maculatus species was more, while in case of Cooch Behar and Jalpaiguri, Calosobruchus chinensis was more abundant. Future scope. The Terai zone of West Bengal is abundant with various stored grain pests due to high rainfall and humidity, which is congenial for growth and development of the insect pest, as well as this region is very rich in biodiversity. So, a continuous research on the stored grain pest identification through morphological and DNA taxonomy may be carried out for exploration of biodiversity and preparation of checklist of stored pest under this zone. Thank you. Stop.